There we go. All right, welcome to microbiology class. And for some reason, I have video this morning. Some reason it just works today. So I'm happy that it's actually working. That's good. What are we doing today? Today we are looking at lecture 11. Lecture 11 is a lecture on antibiotics and life cycles. I'll look at the carbon cycle and the nitrogen cycle at the end of class. And today for questions, I've got 3 people down. I've got Neil, Don, Jackie, and last week, I didn't hear from Sebastian, Robert, or Case. Or Lindsay or Adam, if any of you want to join in with the questions and share your answers with me, that'd be great. Paul says, good morning, I fixed my camera. It's not me, I don't know what happened. So luckily I have the video working today. So let's get showing with the PowerPoint presentation. Share screen. There it is. Okay, so lecture 11, what we're on. So I believe everybody can see my PowerPoint lecture. I don't have a review slide from last week. I'll have to, if any of you have a question, we can just start off right now. Questions, anyone?
She dropping hints?
I hear somebody.
When I read that kind of shit, just go one side of the other.
Israel, we can hear you.
He's like, if we were going to choose.
Hang on a second. There's a setting with my sound. The sound is coming through my computer microphone or computer speakers. I'll set that to the sound in the classroom off the speakers in the classroom so I can hear people if they say something. Speaker sound.
What did he say? You ever heard the story of?
I can hear people on my laptop. I can't hear them on the speakers in the classroom. I thought I would hear them in the classroom. There's people who have got their microphones. Let me look at the participant list.
Matthew, you need to mute.
I've just muted Matthew. There. So Matthew, if you want to say something, go ahead. Let us hear it later. Okay. So my presentation this morning, first of all, discovery of antibiotics. Antibiotics were discovered by, or they've been kind of recognized to be out there. People were looking for something to control other bacteria. And the two major developments last century were by two individuals named Alex Fleming and also Selman Waxman. Alex Fleming was a British scientist from London, and Selman Waxman was from Rutgers, which is in New Jersey. It's just across the border. Yeah, she's on.
I can't join the meeting too.
Okay. I'll briefly talk about multi-drug resistance, especially tuberculosis, and how does multi-drug resistance come up. There's 4 different ways: the bacteria or fungus have different methods of blocking or disactivating something that kills it. And there's also genetic ways that bacteria and eukaryotes transfer information, and that's what I'm going to talk about. And then the final thing is the association with the nitrogen cycle and carbon cycle. Okay, so discovery of penicillin. Penicillin was first discovered by Alex Fleming in 1920s. He was a research scientist who was at a hospital. He was studying bacteria and fungus in the hospital. And there's some mythology about his being an unkempt sort of individual. And this is kind of why he had some contributions to the field. One of his major discoveries was the enzyme lysozyme. And lysozyme, he arrived at thinking that, okay, in the human body, there's ways to prevent bacteria disease, right? Even at the nose, it's like a barrier. How is that possible? And so he dripped some snot or nasal mucosa onto a plate. I don't know if it was him or if a lab technician did this, but they tested the snot and they found out that the runny nose material prevented other bacteria from actually surviving on the plate. And they figured that, okay, there must be something in the liquid from the nose. So they did some chemical analyses and they found a chemical called lysozyme. Lysozyme is a very commonly used industrial enzyme. It's used in a lot of molecular biology. And Lysol, maybe. Oh, it could be a connection. Natalie's saying that lysozyme may be in Lysol. Lysozyme breaks down bonds, so it could be there. But Lysol may break bonds by other methods, like a chemical method. So I'm not too sure if it's an enzyme or a chemical. Okay, so you'll recall from our lab about 2 weeks ago, you did something like this where you had bacteria and you had a disc, and on the disc you measured the zone of inhibition. That inhibition zone was measured, and you try and correlate the inhibition with how active the enzyme is. And that's kind of like the experiment that he did with let's see, I just get a spotlight. He did, so here's a, oops, here's a penicillin colony. And here's some bacteria and a zone. So you measure the distance between the zone to find out how active or strong it is. And that's just what he did. You could also do things like silver nitrate, mercuric chloride, all kinds of different compounds. Okay, so the discovery of penicillin is based on a fungal bacteria. The fungus is Penicillium notatum. And it had grown on a plate at the same time as Staphylococcus. So around the yeast, there was like a zone, and that prevented Staphylococcus to grow. Here, Natalie says Lysol was invented in 1889. Lysosomes were discovered in the 50s.
Lysol was invented before they discovered lysozyme.
And lysozyme, the enzyme.
Sorry. Oh, that was what we did.
And inside of our bodies, we have lysosomes, organelles that actively kill, search out, hunt microbes. Back to the discovery of penicillin. So Fleming discovered this mold, realized that it was effective. He could even dilute, like isolate some of the mold material, dilute it 800 times, and it was still active. And what he could do is he tried injecting it into mice, but it didn't work. It wasn't very good against infections, but he could spray surfaces, and that could work as a topical agent. So he had this topical agent, but he wasn't sure exactly how to get it to work. So it was not exactly the most useful invention or compound for him at the time. He didn't have the chemical expertise to kind of solve what it was. So what do we use penicillin for? Some of you may have had a prescription for penicillin. It has been used for tooth abscesses, ear infection, stomach infection, intestinal infections, kidney infection. It's not used as so much anymore because a lot of people have penicillin bacteria that are resistant to them. And here's my little funny cartoon in the penicillin laboratory. The technician says it was getting moldy, so I threw it out. This kind of describes the case in Fleming's case. Here's a picture of the two individuals that really pushed the chemistry forward. So in the late 1930s, so over 10 years later, these two people, Howard Florey and Ernst Chain, they were two chemists. They were looking for a compound to be active against bacteria or microbes. And they did some reading. They found out, oh, it's already been found. Well, how do we make more of it? So what they did was they have this process where they scale it up, grow it in bigger fermenters, and then in bigger fermenters, you purify it. And I have a YouTube video for us to kind of take a look. Can this compound be made in industry? It is made in industry, industrially, yeah.
Because I saw the chemical form of blood. It contains just CHO and as sulfur nitrogen, carbon hydrogen, oxygen.
Oh, but the chemical formula doesn't describe that. There's also a ring, two rings next to each other.
It's a benzene ring.
A benzene ring and next to a square ring. What is that called? A butane ring? butanol, butane.
Now, that informs my next question. I don't know how the bacteria go about making some of these complex compounds, but you see some other bacteria just make some simple compound. I don't know if there's a mechanism that informs them to make like complex compounds like chains and stuff like that.
Tibetku's question is, how do bacteria make these complex compounds? You think of a microbe as like a factory. What they do is they assemble things. So what they will do is they will, within their enzymes, they will assemble maybe a 6-membered ring and then attach something to the 6-membered ring and then attach something to that. So they're always attaching things to complex molecules. And that's and if they have a mutation within their genome, then they might make something different. And mutations occur at as a rare thing, but when you're talking about billions of microbes, it could happen over time that you get different compounds being formed from these microbes. So it's like a factory, and little bits are attached to them. And every microbe makes a different kind of antibiotic.
So weird.
Yeah. It's so in 1939, during the Second World War, Chain and Florey develop a process, and they actually do isolate penicillin, and it's being used in wounds for soldiers during World War II. And after World War II, it's used clinically, and it's helping people in the public. And because of this, they get the Nobel Prize for this discovery. Okay, so the next antibiotic, streptomycin. So penicillin is kind of like a general antibiotic to cure things like abscesses, skin infections. Streptomycin is important for us because we now have a compound that can cure tuberculosis. And tuberculosis was a really serious problem back in the 1910s, 20s, 30s, 40s. And with the invention of streptomycin, people could control the infection. So we have a way to kind of control tuberculosis being spread around. Selman Waxman is the guy who is awarded the Nobel Prize for the work. I say he's the only person who awarded the prize for the work. And it's interesting because he worked with a student whose name is Albert Schatz. Albert Schatz is the person who actually discovered it, isolated it, and purified it.
So why was he not awarded?
Credited.
He
Because he was a student.
That's the way things went back then.
Tibetku's question is, why is Albert Schatz's name not on the Nobel Prize? And the answer in a lot of cases is they don't award the prize to the people who've done the work. It usually goes to where it was discovered, whose lab it was in. So if in this case, it was Selman Waxman's lab. And this is a question, is that ethical? It's a big question in science.
Well, it's a big part of the reason a lot of Black inventors aren't credited and have not gone down receiving credit
Yes.
for a lot of their inventions. I mean, even recently, like the super soldier, he had to sue to, he just got his payment recently, a few years ago. So it's common practice.
People somehow managed to take invention credit and make money off of somebody else's invention. Yeah. So in the case of Albert Schatz, I got a picture of him here. So he's with his mentor. So these two guys, Selman Waxman, I'm trying to remember his name. Selman Waxman and Albert Schatz are in this picture looking at a tube of streptomyces media. Albert Schatz is the young graduate student there, and Selman Waxman is the older professor on the right-hand side in this picture. What do I want to say about it? So Selman Waxman, like I said, helped develop this into a drug that helped a lot of people for a cure for tuberculosis, endocarditis, which is a heart infection. So if you have bacteria on the heart, it could cure that. So there are a number of diseases that it was good for. Endocarditis is a really hard infection to get rid of because it's in a place you don't typically access easily, and it's anyway. It's
The heart cavity.
yeah.
The-
Bacteria can actually infect parts of the heart. Yeah. Oh yeah.
Yeah, my husband had, he had 2 liters of fluid around his heart. Years ago.
And
Really sad.
what did they use? Do you know if they used streptomycin on him?
They used all kinds of things on him. They drained it. And he was in the ICU.
It's a very difficult infection to get rid of.
Thank God for science, right?
Honestly.
Yeah. The story of Albert Schatz does end okay for him. He finds out that his discovery is making a drug and that it's making his university and his mentor a lot of money. He sues them and
I
he
was
wins
gonna
in
say,
case.
Did he get any money?
He did win and he did get recognition, but he didn't get onto the Nobel Prize. He didn't get the recognition.
You should only mention his name when speaking. Albert Schatz and that other guy.
The professor said there was an alteration to the compound, and that's why Schatz is not on the
I
patent.
say that when I change your recipe and make it my own too.
And the process was patented, but anyway. So what this does, it also comes up with a new research and development. So in a research lab, you spend years trying to figure out a problem, you come up with an answer, and then you take that to a pharmaceutical company, and the pharmaceutical company develops it and make money from it. And this wasn't done before the 1940s or 50s. And this is what's being done a lot now. The models are changing, but anyway,
Started with penicillin.
it started with this kind of drug development.
Or, sorry, what was it?
It's also patents were a big role in this as well. Anytime they have a drug and they modify the process a little bit, they make a new patent. So they have a drug, make another modification, another new patent. And this is a way for drug companies to make money as well. So they control the production, they control how it's made, and distributed. They have all kinds of ways to make money from these inventions. Okay, so here's a couple of YouTube videos I'll share with you guys. Let me know if you can see these. So I go to Zoom. In Zoom, I say share video, or no, share screen. Share my Google Chrome. And here's a 3-minute video on
Until the mid-20th century, tuberculosis, an infectious bacterial disease of the lungs, was one of the world's greatest killers. In the 1940s, a small group of scientists working under improbable conditions in 4 countries pulled off an antibiotic miracle, a combination of drugs that promised to exterminate the greatest killer ever known to man. In the United States, it was Selman Waxman with his colleague Albert Chance. The most well-known of the new antibiotics, derived from microbes found in common dirt, was streptomycin, discovered in 1943 by Rutgers microbiologist and alumnus Selman Waxman, graduate student Albert Schatz, and others. Streptomycin would become the first cure for tuberculosis, then the world's most dreaded disease. In 1900, more than 147,000 people died from the disease annually in the United States. By the early 1960s, tuberculosis deaths dropped by more than 90%. Studies by Waxman and his group on actinomycetes found in soil would ultimately yield more than 20 antibiotics. Of his discoveries, Waxman would famously say, "Out of the earth shall come thy salvation."
You know, it was a very funny thing. When I received an hotel flight, 1952, that was in October, you can well imagine we lived in the Brunswick, I was connected to the structure, in a whole class of reporters and commentators from came down from New York to interview me. So they said, all right, give us a quotation. What was your life represent? So I said, well, my life represents an ancient saying, out of the earth shall come thy salvation. And that was that. And they published it. Within 48 hours, I began to get telephone calls from various magazines like Newsweek, Time. Where did you get that quotation? You looked also the Bible and you can't find it. I still think it's fair to put on the Bible. I made it myself on this corner of the moment.
Waxman would appear on the cover of Time magazine in 1949 with this quote, "The remedies are in our own backyards." And in 1952, he awarded the Nobel Prize in Physiology or Medicine. With royalties from his discoveries, Waxman's Foundation for Microbiology supported the establishment of the Waxman Institute for Microbiology, which thrives to this day.
So that's the video for Selman Waxman. And I have a video on, let me just check in class. Natalie, did you see that video on your screen? Did it show up?
I was watching.
Oh, you're watching here, okay. All right. I was just thinking, did it show up for everybody? Let me just ask out there, everybody, did everybody see
I
it?
do see the YouTube.
Okay,
Just like that.
here's the next one.
Paul says the sound is a little slow. They did see it.
You did see it. Here's, this one's a longer one on penicillin with Alex Fleming.
And disease. These three ancient enemies of man still stalk hand in hand across too much of the world. Of the three, disease can be said to be the most deadly. Disease often fathers hunger and poverty. This is the story of how one of the mightiest weapons against disease was forged. It is the story of penicillin, the first of the modern wonder drugs known as antibiotics.
It's a longer YouTube video than I wanted.
Today, the penicillins are only one group of antibiotics among many, and in laboratories throughout the world, the search goes on for more. To learn how the first of them, penicillin, was discovered and developed, one must know of the work of 3 British scientists. What they achieved has revolutionized medicine. Sir Alexander Fleming, for the initial discovery of penicillin, Dr. E.B. Chain, and Sir Howard Florey, for their finding its curative power to benefit millions. Early in the 1920s, in London, Alexander Fleming, a young bacteriologist, worked in the inoculation department of St. Mary's Hospital. Familiar with antiseptics and inoculation as weapons to help the human body battle against disease germs, he began to wonder what natural defenses might be possessed by exposed parts of the body, such as the eyes. In each place, Fleming found a protective substance which could destroy some germs before they invaded the body. Some germs, but not all, and not the most deadly.
Lysozymes
In one of the
protective.
glass dishes where he cultured
barrier.
germs for his experiments, Fleming noticed one day in 1928 that some mold, such as appears on decaying food, had begun to grow. A spoiled experiment. But with mind alerted by the earlier work on protective substances, he looked closer. He saw that near to the mold, no germs were growing. Might it be that this mold, like the human body, produced a substance capable of destroying germs? He made tests and found that even some of the most dangerous germs died near to the mold. He then grew the mold in a meat broth liquid. The color of the liquid changed finally to a bright yellow. This liquid proved lethal to germs, even when greatly diluted. Not pure enough to be injected into a human body, it was nevertheless effective on wounds. Fleming published what he had done, calling his substance penicillin. Attempts to purify it and to extract its essence failed. In the 1930s, a new chapter opened at the University of Oxford. The professor of pathology, Dr. Howard Florey, was joined by Dr. E.B. Chain. Together, they planned a research project on natural germ-killing substances. They unearthed the now old papers on penicillin. They formed a team. The great quest was on. First, Chain and his colleagues set to work on the chemical problems of extraction and purification. They found a way of transferring the active penicillin from liquid to liquid until it was held within almost pure water. Which had a brown pulse. Here was a powerful form of penicillin, still crude, but which produced no ill effects in animals. Florey prepared the great test. The germ streptococcus is a killer. This he injected into mice. Half of them then received the brown powder. Those who had been given the penicillin lived and were healthy. And Professor Florey declared, "It looks like a miracle." But attempts to make it pure enough for man caused it to lose its potency. Chain and a colleague, Dr. Abraham, then devised another method. Dissolving the crude penicillin in ether, they passed it through a long tube filled with alumina, each substance producing layers of differing colors. In the yellow layer was penicillin. They repeated the process again and again.
It's a lot of work, yeah.
Now Florey could try penicillin on human beings. The results proved the healing power of penicillin. But by now, World War II was raging. The need for penicillin's healing power was enormous for soldiers and for civilians. But in 1941, purified penicillin was still desperately short, and Britain was hard pressed by air attacks. To make the quantities needed, it was decided to seek help in the United States. Here, scientists devised better ways of culturing penicillin, which could give much bigger yields. Production in quantity could now be foreseen. With encouragement from their government, American drug companies tackled the difficulties of producing penicillin on an industrial scale. Florey returned to Chain and the others, able to look forward to supplies for further research. Alexander Fleming had a patient dying of meningitis. Fleming turned to Florey, and Florey gave him all the penicillin that could be made available, and with it, his advice. Profoundly impressed by this personal experience of penicillin's dramatic power, Fleming addressed himself to the British government about the need for large-scale production. And the press took up the cause.
I think I should pause your quote there.
The big British chemical and drug companies and the research workers now also projected the big-scale production of penicillin. The mold was grown in thousands of flasks.
There's the bacteria part. Okay.
Britain's output joined the huge supplies now coming from the United States to give life to hundreds of thousands of soldiers. By the end of the war, flasks had been replaced by great steel vats, each making 15,000 gallons of penicillin liquid. And huge factories were in production both in Britain and the United States. Penicillin was now for the world.
Okay, I'll pause it there. It's just about the whole thing. Back to Zoom. Thanks, Will. Let's stop the sharing now. There, we're back to the video. Okay, so as you can see, penicillin is a compound that you have to grow a lot of bacteria, or in the case of streptomycin, you have to grow a lot of bacteria. In the case of penicillin, you have to grow a lot of fungus, and you have to grow it in great big quantities. And there's so many steps to figure out before it becomes a healthy thing you can add or give to people. So it's a laborious process to do. Great. Let's just take a look at the PowerPoint again. Where are we? Here. Okay. So penicillin is invented, streptomycin is invented, and we have a couple questions. Let me just take a look at the participant list. It's turn for Neil, Don, and Jackie. Neil, are you in? First question. No Neil today. Don, are you online? Don. Question for you. Who discovered penicillin? Can you tell us?
Credited.
Who is credited for discovery of penicillin?
Because we know you discovered it.
It's
It's
a
a
lot
different
size.
question.
It's a lot
It's a
size.
different question.
Who is, who actually did discovery? We're not exactly 100% sure, but yeah, who is credited? Thank
It's
you.
one by a lot size scientist.
Exactly.
A lot technician.
Or Jackie, Don or Jackie.
Tell you she's not near her computer
Oh, not near a
at the
computer.
moment.
Okay, Jackie, how about you? Are you close to a computer?
To Matthew.
Oops. Oh my goodness. How do I get rid of off of Matthew?
Direct message people in the chat.
Everyone.
Want me to help you out, Andrew?
There we go. Okay, Natalie's going to answer.
Okay. Who was credited for the discovery of penicillin? Selman Waxman was credited.
No?
Wasn't he credited? Oh, wait. Oh, I'm on streptomycin, sorry.
Oh, good.
Penicillin, discovery by Sir Alexander Fleming.
Thank you. And what disease can be treated with penicillin? What infection? Streptococcal infection. Okay. What organism is penicillin from? What's the organism name? Do you guys remember?
It's a fungus.
It's a
It's
fungus.
a fungus, it's a mold.
Penicillium.
Thank you, Brooklyn.
What is it?
Penicillium species or something.
Penicillium notatum. Yeah. And what type of organism makes the antibiotic streptomycin?
Bacteria.
It's a bacteria. Good. So the penicillin's made by a fungus, streptomycin's made by bacteria. What disease can you treat with Streptomyces griseus with those antibiotics? Tuberculosis being the big one, yeah, because tuberculosis was a bad disease, fatal disease for so many people. Okay,
How was penicillin discovered? How is that answered?
I didn't answer how was it discovered,
It
but
was from they went on and found it and left.
Oh, that
agar
part.
plate.
Okay.
Yeah.
Okay.
So it grew on an agar plate of something that goes and kills some of the stuff.
Yeah.
And the codex,
okay, thanks. Questions have been answered here. I don't know, do you guys hear those answers? Tobachku answered that how it was discovered. It was discovered accidentally. A plate with bacteria on it got moldy, and the mold prevented the bacteria from growing. That was the discovery. Okay, so the next topic I've got is multi-drug resistance. And the one disease I'm talking about is tuberculosis for a bit. So in the 40s, tuberculosis had been decreasing, and they're very happy that they found a drug to cure tuberculosis, because tuberculosis can be lethal 50% of the time. But if you take streptomycin as an antibiotic, it takes a long time for the infection to cure because it's so, biofilm in your chest is so difficult to remove the bacteria. It takes a lot of oral antibiotics to get rid of it. Now, instead of a 6-month treatment, you can use other antibiotics, rifampin. Here's the 6-month treatment, rifampin, isoniazid. So there's a couple of different kind of antibiotics now that are available for 2 months, and then rifampin and isoniazid for 4 months. So it's now like a 6-month treatment. But it's still a very large killer. As you can see here, for all forms of tuberculosis in the world. There's like 9 million cases a year. 1.5 million people are still dying every year from tuberculosis. And that's broken down into people with HIV as well. So every year, 1.2 million people with HIV contract tuberculosis, and 400,000 of those patients die. With multi-drug resistant tuberculosis in the world, there's 500,000 people that get this, and 200,000 people die every year. So there's a drug resistant strain that's killing people out there every year. And there's even a new strain, extra drug resistant tuberculosis. And we don't know how many drugs or deaths are attributed to that. So every year, there are people getting diseases, tuberculosis. So they're trying to figure out strategies to reduce the number of deaths. So the focus, they're thinking, shouldn't be more on preventing this disease. You may have heard there's a, what is it? A BCG vaccine. The BCG vaccine is a vaccine that can be administered, and this will prevent tuberculosis, but it's not used so much in Canada or United States. It's used in other countries. So there is a vaccine for tuberculosis infection. So how did we figure out there's something called antibiotic resistance? So back in the 1940s and 50s, you have these drugs, streptomycin, chloramphenicol, tetracycline, and they started using them. When you use a chemical to treat disease, that's referred to as chemotherapy. So chemotherapy started being effective against diseases, and it was effective against gram-positive bacteria, gram-negative bacteria, and parasites, and tuberculosis. And they're using things like sulfur drugs. Okay. Oh, look at over here. I have a sign, 1950 sign by Bonds, 1940s. Penicillin cures gonorrhea in 4 hours. Wow. So if you had a gonorrhea, antibiotics act fast. So they do start killing really, really quickly. And gonorrhea and syphilis are two diseases that we don't see them so much anymore, but they are still out there. Some people don't take care of themselves. So I was saying, you have chemotherapy fighting diseases, but people were coming to hospital and they're having these infections, and they were not responding to the drugs. And so they're wondering, like, why is it that these people are not responding to the drugs properly? They found that sometimes bacteria develop a resistance to drugs. And I have here 70% of bacteria that cause infections are resistant to at least one of the drugs that are used against them. So they have all different kinds of ways of protecting themselves. In fact, 1946, the very first report of antibiotic resistance is reported. So somebody developed a resistance to, had an infection, and that infection wasn't treated with streptomycin. Here's an instance, 1953, Japan, there's an outbreak of dysentery, and they find out that this dysentery is drug resistant. So we've known about drug resistance for quite a while. How does this happen? Well, bacteria have a way of defending themselves. They have natural systems, and I'll talk about some of them, and I'll show you a couple. If they have a system for getting rid of an antibiotic, that could help them. If the organism doesn't have a antibiotic target, so some antibiotics target the cell wall, and the cell wall has special proteins and decorations along the cell wall. If the antibiotic doesn't target the proper compounds, the bacteria is going to survive. And as you know, the outside of the bacteria has got this cell wall. It's very tough cell wall, and that's impermeable to some antibiotics. Bacteria can also do this thing where they gain resistance. So what they'll do is they'll borrow from other bacteria, resistant parts of other bacteria, and use that. It's I'll talk about that in a bit. So bacteria can also do this thing where they chemically modify the antibiotic. So Bachpu noticed that the chemical formula is fairly complicated. And he mentioned that there's also like a benzene ring within the molecule for penicillin. Microbes can attack part of that ring structure. So there's two rings together, like a 4-membered carbon ring and a 6-membered carbon ring, and they can attack the less strong ring. And if they do that, then they inactivate the antibiotic. There's also a film that bacteria make, and this bacteria is referred to as a biofilm. But antibiotics have a hard time getting through biofilm.
Is that within your gut?
That's anywhere there's bacteria. So you can call the kombucha layer a biofilm. You can call slime on top of a bathtub biofilm. Things within your bar, like if you're serving drinks at a bar, they have to remove the biofilm and clean out the biofilm because that can cause customers to get sick. So biofilm exists everywhere in different forms. So it's like a slime layer that certain bacteria make. And these layers, the biofilm layers, prevent bacteria from being exposed to antibiotics. So there are, pardon?
They can hide.
Yeah, it's like a hiding zone. Good.
I watched a PowerPoint about like a doctor. She was presenting. She has her own kind of supplements and whatnot to for gut health, and she was talking about biofilm that can cause infection and disease within the gut, and you don't know it because you can't.
You don't see.
Excrete that on your own. So she was just different supplements can help you get
Can assist, yeah. There are chemical signals for microbes to tell the microbes to disperse, like they can form a biofilm, then you can send a chemical signal, and the chemical signal will tell them to disperse, and they'll leave. But what is that signal? That's an active area of research. People are looking at that all the time. Okay, so questions for the class? Oh, Amanda's got a question about gonorrhea.
Of course she does.
Because it's a fast-acting antibiotic. You catch it in the morning, by supper time, you can have it treated. Yeah, thanks. Very fast acting. Okay, so how common is drug-resistant tuberculosis? And as you saw from the slide that I showed, it's fairly common. It forms a percentage. I don't know, was it 5% or 10% of the total population? 70%? Question I had, does anybody here know someone who is affected by drug-resistant infection? And if you may have met somebody who's had one and not realized it, or somebody may have had one and been really drastically affected by it. It's a real serious concern in ICU units, that's for sure. Yeah. Brooklyn's got a question. Go ahead.
How would you tell the difference between that and when your immune system attacks itself? Type of disease?
Immune system attacking itself. I've heard of this. I'm not exactly sure.
Yes.
Immunocompromised, yeah. How's that different? So drug resistance.
Drug resistance compared to immunocompromised.
Yeah, immunocompromised is the host being affected by the disease. And their response is compromised somehow, it's not acting properly, and the host is their system is acting on themselves, so, as opposed to acting on the disease. So that's kind of what happens there. I think that's from my understanding. It's kind of like a rough understanding.
I understood immunocompromised is like someone whose your ability to fend off something being compromised because you are not 100%. Because again, like the way you explained it, maybe, but is it necessarily because something's fighting against you?
Definitely immunocompromised, you're always fighting something. And for some reason, there's some sort of feedback where the feedback within the person is not acting properly. Yeah.
It's a scary thought, you're always fighting something. It's true, right?
It's kind of true. Our immunity system is always there. It's always responding to threats. And Matthew sent me a message. He says, I think we've all been affected by drug-resistant infection this year. Well, that's in a way, yes, it's very true, because we didn't have a way to fight off COVID infection before last year, well, other than natural immunity. Okay, so that's drug infection. Here's a little bit more information about those: chemical removal, modification, blocking, and gene transfer. And there's 3 kinds of gene transfer. I'll just go through this fairly quickly and then get on to the nitrogen cycle. So here's the mechanisms. As you can see from this slide, you've got a blocking effect. So here's an antibiotic. So something's blocking the entry. And I was talking about how you could have a biofilm or cell structure. They would block your antibiotic to getting into your cell. A second way, you have enzymes inside of the microbe. And what the enzymes do is they act on the antibiotic and break it down so the antibiotic's no longer effective. So that's a chemical inactivation. Third way, so a cell has a natural target for an antibiotic. What the cell can do is modify its own interior, change the shape of the interior structures, And by changing these interior structures, it alters the target. So the penicillin or the whatever antibiotic isn't effective against that structure. So the cell can actually kind of alter the molecules that are being targeted. And the final way I've got here is efflux of antibiotics. So this is another, these four fields are fairly large fields in microbiology research, where you're looking at the pumps of microbes and how effective pumps are at getting rid of antibiotics or other compounds. There's four mechanisms. I talk a little bit here about biofilm being like a consortium. It's a whole bunch of microbes that live together, and it can be different kind. It can be like the mother of liquor or the mother from kombucha. And if you think about it, here's a biofilm. They have like a free swimming form. Free swimming form attaches, and then once they attach, they form like a group or a colony or a structure, and the structure eventually becomes a film with a whole bunch of microbes in it. And if you somehow treat this big biofilm, there might be some sort of dispersal agent, tell the bacteria to disperse, they will. They can do that. It can be a drug. There's no really good drugs at the moment for dispersing biofilm, but it's an active area of research. People are looking at it, trying to figure it out. They're coming up with ideas.
Would you trust supplements online?
I could try it. The question here was, would you trust supplements? You could try it, see if it helps.
Seemed like she knew what she was talking about. So.
Yeah. Here's another slide about antimicrobial resistance. So if you think of bacteria having resistance, maybe about 1%, what happens is the 1% can increase because bacteria or small microbes can exponentially grow and share information. So even after 10 years, after use of a compound, strains can become resistant to the drugs, which can be a problem because then at some point there's no ability for you to treat the drugs or treat the infection with drugs.
Are they working on this right now?
Yeah. Yeah. Active area of research. So there's different words for how this information is transferred within microbes. There's something called vertical gene transfer. So I'm saying here, it's kind of rare for microbes to have a resistance, like develop a resistance, like 0.0000 with 8 zeros and a 1. Like that's how often it happens, like 1 in 100 million times. Because they do mutate and the mutations are rare, but because they grow so fast, then you can get a number of cells that get this resistance. Here's a picture for vertical gene transfer. So the genes are being transferred directly to other organisms. There's also something called horizontal gene transfer, where an organism dies and that DNA is out in the environment or transferred somehow in the environment to another bacteria, and the other microbe picks it up. So there's vertical transfer and horizontal transfer. Yeah.
That's weird as hell. Like, what is the, how are they able to do that?
I'll show you. Will's saying this is very weird. And yeah, I've got a couple of slides on this. So horizontal, like I said, transfer between species. There's 3 different ways to transfer this DNA. There's one where 2 microbes can just park next to each other, attach to each other with pili, and exchange DNA through the pili. So that's called conjugation. And in conjugation, they have like small circular chromosomes that they can share through these pili. So DNA goes back and forth this way. This happens in E. coli. And this is a very useful technique in E. coli because E. coli can conjugate and transfer DNA back and forth like that. There's another event called transformation. In transformation, the DNA is in the environment and the microbes pick up the DNA from the environment. How do they do that? One way is chemically. They encounter a chemical that loosens their pores and the DNA just kind of slides in and they get that DNA. Another way is by electricity, where electrically they are stimulated somehow and the pores are loosened, relaxed, and this DNA comes in. So there's different ways to transform microbes. And then the third way I've got here is transduction. And by transduction, this word means where a virus comes and shares the DNA into a microbe. And here's 3 pictures. Here's transduction. So you got a virus with some DNA. It goes to another microbe and transfers the DNA to the second microbe. Conjugation, where there's some DNA exchanged through a pili, and transformation, where a microbe is killed, that DNA is in the environment, and it's picked up by another bacteria. So there's different ways that they can pick up this resistance. Those 3 words would be words I'd probably ask you about. So transduction, conjugation, transformation. So just know those 3 words. So what are some ways of getting us? Why is this important to us in food science? Well, we use antibiotics on animals. We use antibiotics with chickens, cattle, pigs, and those antibiotics that can find their way into municipal water because they go into the water stream. So it's a double hit. You can get antibiotics in your food and you can get antibiotics in drinking water. I've seen studies where what they do is they take fish raised at a fish farm and they expose the fish to antibiotics. And then they take a little bit of the flesh from the fish that's raised on the fish farm and they put it on a plate and see if that actual flesh is got a zone of inhibition, and it does. So it can exude the antibiotics even after it's, yeah. So, and that's a complicated problem. Is it from the fish flesh? Like, are there natural products in the fish flesh that produce this, or is it from the antibiotics? But that wasn't answered for me directly, but I have seen people try to show that. So some of our foods are being exposed to antibiotics, and it's kind of routine in North America, but in Europe, they're trying to ban antibiotic use. Just to say different areas treat antibiotics differently. So in Europe, like I said there, many industrialized countries are trying to stop the routine feeding of antibiotics to animals. Non-therapeutic use of antibiotics in livestock is 60% of antibiotics in the United States. If you think about that, this is non-therapeutic. What they're doing is they're putting the antibiotics into the feed to get bigger animals. So it is a way to develop animals. So this also may increase with antibiotic resistance as well. It is a concern, but It's a practice too, yeah.
Almost makes too much sense.
Go ahead, question.
Just wondering how it would make them bigger.
How does it make them bigger? Well, I'm not 100% certain, but it may inhibit certain microbes that are naturally there. Because if you feed an animal some antibiotics, then they have less microbial strain on them, bacteria strain, some sort of strain, and so they might end up being healthier, bigger, stronger. And there's also genetically modified crops that have antibiotic resistance genes in them, herbicide resistant genes or insecticidal genes. That I just mentioned that there. Okay, okay. And there's two questions here. Question one, name 4 methods used to block antibiotic activity, and how the 3 processes of transduction, transformation, and conjugation work. So those 2 questions. Think about it. Can you answer the questions? Cass says, it's a way to keep animals healthy. Thanks, Cass. Healthy animals grow better. I've moved it off the screen. Healthy animals grow better and do better. So for things like colds for animals, colds for humans, it's like for young animals, you give the young animals some antibiotics to, they do better. And Brooklyn's got a question.
I was just gonna answer the question.
Oh, go ahead.
I don't
Answer the
know.
question.
So for antibiotic resistance,
Name 4 methods. Yeah.
lock entry,
Blocking entry. That's definitely one.
inactivating enzymes,
Chemical modification, inactivating enzymes.
removal, efflux of environments. What is efflux?
Efflux means sending it out, pushing it out. So it's like an efflux pump. You're effluxing the antibiotics.
And modification of target molecule.
Great. There's target molecules inside that are altered by the microbe, and they do that naturally. And if they can do that, then they've provided a resistance for themselves. So you guys want to try the process of transduction, transformation, or conjugation? Somebody, anybody? Horizontal gene transfer. Horizontal gene transfer, if you have them. Can you see that? You have a bacteria that has some DNA inside. This bacteria dies. So the cell wall is out there and it degrades, but the DNA is picked up by a bacterium that doesn't have it. And so that gives the resistance. This is known as horizontal gene transfer.
What picks it up and what does the same thing?
Yeah, so it uses this useful DNA piece to have resistance to an antibiotic. What's that?
Those guys are crazy.
Bacteria are crazy. They try and survive somehow. It's a war out there for the bacteria. Yeah. Transduction, if you remember, was the way that DNA is shared from a virus to another microbe. So a virus can pick up some DNA, take it to another microbe, and infect the other microbe with that DNA. That's transduction. Transformation is the either electrically or chemically where you have horizontal genes transfer, and conjugation where two microbes sit next to each other, park next to each other, and transfer DNA through a pili of some sort. Okay, next. We're off onto the plant and animal cycles, nitrogen cycle and carbon cycle. Okay, so that's it for antibiotic talk. And I'll just talk about how plants and animals have, how they take up nitrogen and how they take up carbon. I wanted to also talk about the assignment at the end of lecture. So I'll watch the time, try and give myself 10 minutes to do that. Okay, so if I have to cut off the carbon cycle, we can pick that up next time. Nitrogen cycle. So nitrogen, where does it come from? Air. Good. Nitrogen is found in the air. It's abundant in the air. And what plants and animals have to do is they have to fix it. And by fixing, I mean they have to modify it somehow. So here's an image. Your nitrogen is in the air. And then you have bacteria of some sort, cyanobacteria, legumes. Legumes are really important. Legumes like peas, beans. We follow these fairly closely. And these organisms, what they'll do is they'll take the nitrogen from the air, convert it, and when they convert it, animals can feed off of it, or the plants break it down into a form that can be, when it's in the soil, can actually be used by other bacteria. And this gets converted into ammonia, which is then very useful for the soil. So on my wheel here, we have nitrogen. It can be converted into NO2 minus. Oh, sorry, NO2 minus can be converted into nitrogen gas. Nitrogen gas is converted into NH4 plus by a diazotrophy method. This is ammonification. So the nitrogen gas is broken down and hydrogen is added to it. And if it becomes NH4 plus, then it can easily become NH3, ammonia. And from ammonia, it can become other compounds, including, again, if it's oxidized to nitrogen gas or nitrite, or if we further change nitrate, nitrates can go to plants, ammonia, and so on. So it's like a whole cycle here, where it's going from the air to the plant, decomposing to the bacteria, back to the plant, decomposing bacteria, maybe nitrification, back to the plant, to the animal, and so on. It's just being cycled around and around. The last lab in our assignment was supposed to be looking at a microbe from a plant. I canceled that lab because our first lab was canceled on account of the storm. So we're not going to take the pea plants and crush them, but this is something that is done in the microbe labs. We can take a pea plant, look at the roots, the roots have little red nodules, and it's red because it's got iron in it, and the iron oxidizes. And if you crush that, you can see the microbes that are fixing the nitrogen. Nitrogen is important because we need it in our cells, we need it in our DNA. And so there's lots of different kinds of nitrogen metabolism. There's nitrogen fixation, anaerobic respiration, and nitrification. Let's just go through those three terms. So nitrogen fixation. This is what happens when you convert ammonia gas, is you're fixing the nitrogen into NH3. So I just got to move this. So it's being converted into NH3, which is ammonia, and the ammonia is then assimilated into a plant or another organism. So this is a way that nitrogen gets into soil and it gets into plants, is by nitrogen fixation. So it's really important that living organisms do the nitrogen fixation, because then you have a useful form of nitrogen. When you have crop rotation by legumes, you're introducing healthy nitrogen into the soil. What kind of bacteria do this? There's Clostridia, Azotobacter, Cyanobacteria, Rhizobium, Frankia, the different kinds of names of bacteria. And inside of a plant, like I was saying, you have these red nodules. The red material is referred to as leg hemoglobin. So it's a special form of hemoglobin. It's a red liquid that is formed by these bacteria in the root nodules. So that's nitrogen fixation. Here's a picture of what this looks like. So on the left-hand side, the gray SEM picture shows these bacteria on the root nodules. And here's an actual root with the nodules sticking out of the root. That's kind of what the pea plant roots look like, is they got these little red bumps. And what's going on is you have the nitrogen fixing bacteria forming in these nodules of these leguminous plants. So on the left, nitrogen fixing Rhizobium bacteria colonize on the root hairs of clover plants. On the right, Rhizobium bacteria on plant roots. In the nodule, the bacteria fix nitrogen. So it's an exchange. The bacteria fix the nitrogen, the plants use the nitrogen. So yeah, Will's got a question.
Maybe you'll cover this, but in like the nitrogen cycle, I've heard that nitrogen is very unstable. So it, I guess, it like breaks down and this again. And is that how it's reacting to the atmosphere?
Yeah, so if it's in the NH4 plus, it's kind of reactive, and then breaks down to NH3, ammonia, which is the smell from the amine smell from nitrogens, and as you know, it's very strong smelling and very volatile. So it does break up, and if it's in the environment or in the air, it breaks down, it oxidizes and becomes NO2 or just nitrogen gas. So it is fairly easy to break down. Hard to kind of form it into NH4, but once it's NH4, it's very useful. And everything uses it. Everything uses nitrogen. Here's a rhizome bacteria. So we're going to miss lab 11, but I'll make up a lab tomorrow. It won't be the same. It won't be, I think it's good, but I've got an idea for lab tomorrow to do. So atmospheric nitrogen goes into your plants. It's assimilated, and this can be your legumes, different kinds of plants, and then it's decomposed into ammonia, and it becomes nitrates, nitrites, and bacteria use it here in your legumes. So if you take the little red nodules, crush them, you can look at them under the microscope and see that there's bacteria there. Yeah, question from Brooklyn.
This is the same thing. Nitrates, are those in food products?
Oh, yeah. We have nitrates of some sort. What's that? Preservatives. So nitrates are often found in meat products as a preservative. Nitrates, nitrites in some form. Nitrates are the chemical nitrates are kind of frowned upon a bit by meat industry now because some people have health problems from too much nitrate consumption. And we have healthier, what we think are healthier nitrate sources, like celery is being used as a nitrate source by Maple Leaf Foods.
Yeah, I saw those and I was like, what even is that usually when something sounds?
It's used as a preservative to help us maintain the stability of the meat, usually. They add nitrates. How are nitrates associated with animals and plants? Nitrogen products are beneficial and everything uses it. Insects, invertebrates, fish, animals, plants. In order for microbes to use it in intestines, they digest nutrients and they make growth factors as well as vitamins. So this is where we get our vitamins from, is from some sort of nitrogen fixation. Here's another example. Plants like peas, clover, alfalfa, they live closely to bacteria and they extract nitrogen from the atmosphere and they supply nitrogen for plant growth. So this is why farmers use alfalfa to recover soil, is it imports a bunch of nitrogen, fixed nitrogen to the soil. So it's very healthy for the soil. Clover, beans, peas, alfalfa, very healthy for the soil. Nitrification, this is the opposite of denitrification. Nitrification, denitrification. It's referred to as ammonia oxidation. So in this case, your oxygen replaces hydrogen in nitrification. So the other term was nitrogen fixation. This is nitrification. Here you have ammonia and the hydrogens are stripped off of the nitrogen and oxygen is attached. Nitrifying bacteria like Nitrosomus utilize ammonia as an energy source. And another bacteria called Nitrobacter will change NO2 to NO3. So nitrite to nitrate. So even adding more oxygen to your nitrogen. Nitrifying bacteria happen in aquatic environments. So they are found in ponds or other places where you have lots of water. So something like a Nitrosomus can convert to nitrite, and then your Nitrobacter converts it into nitrate. So you can see there's NH3, ammonia, converted to NO2 minus, NO2 minus is converted to NO3 minus. So it's making these less usable forms of nitrogen in the environment. Here's where is the nitrification in this? Right here, there's NO3 minus, NO2 minus, nitrates, nitrates. denitrification, it's important for groundwater to be a part of the process. And this can be anything, eukaryotic bacteria, archaea, and NO3 has one of the oxygens removed to NO2. So, and then to either nitrous oxide or nitrogen gas. Use of oxidized forms of nitrogen can also be formed for electron acceptors, so cells can use these compounds. It says here, bacillus, the majority of microbes that denitrify are facultative microbes, so they can exist with or without oxygen. We have bacillus here. It's a common soil inhabitant, and it uses nitrate as an electron acceptor. So it uses it for its cellular processes. Anaerobic denitrification is also a process. You take nitrate, and it converts right directly to nitrogen gas. Now, NO3 minus is a really difficult fertilizer to obtain, but it's an important fertilizer. And one of the reasons that we till soil is to kind of introduce a little bit of air to the soil. And what that does is it preserves the nitrate fertilizer in the soil. So there's a reason for digging up your dirt, and that's to add a little bit of air so that the nitrifiers can get some activity going. So this is why we, in the spring, we dig up the garden, dig up your fields. It's to provide air to these microbes. Okay. And then there's a chemical process called the Haber-Bosch process. Haber-Bosch are the inventors of taking hydrogen gas, nitrogen gas, and they convert it into ammonia. And this is a very expensive, large chemical process now being taken everywhere in the world to make liquid ammonia gas, liquid ammonia. Industrial method for fixing nitrogen uses high pressure, high temperature, uses lots of energy, generates a lot of waste. And that's my spiel on nitrogen fixation. Okay. I know there's only 10 minutes left. And I do want to talk a little bit about the assignment. So the next part was talking about the carbon cycle, but does anybody have other questions? I've talked about nitrogen fixation. What is nitrification? And what is denitrification? Yeah, the soil nitrogen, the ammonia, NH4 plus. That's the nitrogen fixation. And what was nitrification?
Converting ammonia to nitrates or nitrites.
Yes, okay, good. And I think that's all the questions I'll ask on this material, okay? I have 10 minutes. I want to share with you guys the assignment. So my final slide here. Assignment number 5, I ask about selective versus differential media. And I'm going to just share this. Oops. Stop sharing, stop sharing. I'll share something else, share my Word file. Microsoft Word. So everybody should be able to see my Microsoft Word file. So the Word file is media descriptions. Like we learn in class, microbes require media to grow, and you have different kinds of media out there. I'm going to ask you to use the manual to identify some things. So the first question is identify a media that is selective for food-based bacteria. So when I say food-based bacteria, it could be like a pathogen or it could be a beneficial microbe. It's up to you. Just take a look at the description, try and find the name of the microbe. So here I have the first question. I've picked Staphylococcus aureus. I don't know if you see that. How did I pick that? Well, I went to the file, the PDF file of all of the media, and I just did a search for 2 words. So when you're doing the search, look for selective or differential. And then figure out if it's bacteria or eukarya. So I'll go kind of show you what I mean here. And here I have what is the name of this media for Staphylococcus? What is it? What makes it selective? Now this I copied directly from the file. Baird Parker agar contains peptone, beef extract, carbon sulfur. Sodium pyruvate is in the media, and that stimulates Staph aureus. Tellurite additive is toxic to egg yolk, and so only Staph aureus lives. Okay, so let me show you that. We'll go to my PDF. Here's the PDF of my PDF of the media, and here's a nice little picture. Take a look. Here's E. coli in this media, and what's in this media. If you add lysine, it's pink compared to Shigella, which is yellow. So here's uninoculated, it's pink. And then if you add Staph Salmonella typhimurium, it turns no difference in color change, but the bacteria is growing. But Proteus vulgaris, look at this, is a very differential kind of media. Anyway, so if I go into the search term here, okay, I had Baird Parker.
You're just searching this document and filling it in, right?
That's right. So I said Baird, B-A-I-R-D. That's why I can't find it. B-A-I-R-D.
Well, you have this in front of your stuff.
I don't see my search. Oh, diff. Geez. B-A-I-R-D. Oh, there we go. Here's a Baird Parker. And just to show you what it looks like, here's a Baird Parker plate. So uninoculated, take a picture of it, uninoculated and inoculated. Okay, so when I say look for the 2 terms, so selective, now there's lots of 257 selective, and just kind of go through the word selective, find a nice picture. Bum, 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 bum. This word selective shows up a lot. Oh, there's a nice picture. Hey, here it is, a pretty picture. So this BCYE agar, Legionella. Okay, so Legionella is not a great bacteria for food, but it is involved in the food industry. Legionella is found in that air spray that goes over vegetables. They find some people get Legionella disease from that Legionnaire's disease. Yeah. Anyway, so there's a picture for you. So just keep going. Look through the file for selective. Oh, here's another picture. What's that? And just pick a couple of good pictures based on the word selective or based on the word differential. Okay. And how do you get the name of the bacteria? The bacteria's name right here. So copy paste the name of the bacteria. You'll have to read how it's active, like, and copy that little paragraph for how it's active. Okay. So just go through the file, find the information for the name of the bacteria, a picture of how it's what the plate looks like, and do this also for differential. So selective, if, how do you spell differential? There. Let's go down here. I see a picture. Oh, there's one. Here's another one. Oh, that's nice. So, and we could use here Biggie agar. Biggie agar is for candida. So there's a fungus. So half of the assignment is for bacteria, half the assignment is for fungus. Candida is a fungus. If you're not too sure if that microbe is a fungus or bacteria, use Wikipedia just to confirm.
We're just filling in the form.
Just filling in the form, okay?
Always up here.
Fill in the form of the names of the microbes, a picture, copy the picture. Okay. If you need more, I said this Friday, if you need more time than this Friday, like next week Friday, I'll open it up for next week Friday, but that should be it. Okay. I'll make it to next week Friday. So is the form posted? The Word file should be there. Can you find the Word file?
I think it was in the assignment.
It's in the assignments, I think.
But it's not in the content.
I'll put it into the contents. Okay, at the end of class here. And that's it for class. Okay, guys. There's another group waiting for us to come in. Thanks. There is one other thing I wanted to mention. There was a question about the final exam being on the Monday. I can make the final exam go from Monday around noon to Tuesday around noon. So that next week, 18th. Okay. And it's on the PowerPoint slide. Thanks, guys. We'll see you guys in lab tomorrow. Good luck. Thanks, everyone, for being online. Have a good day. We'll talk to you later. Cheers. See everyone tomorrow. Cheers.